In continuation to the previous lecture, today we will start irreversible and reversible inhibitors. Irreversible inhibitors. They are covalently bind to an enzyme, cause chemical changes to the active sites of enzymes and cannot be reversed. A main role of irreversible inhibitors include modifying key amino acid residue needed for enzymatic activity. They often contain reactive functional groups such as aldehyde, alkenes or phenyl sulfonates. These electrophilic groups are able to react with amino acid side chains to form covalent adducts. The amino acid components are residues containing nucleophilic side chains such as hydroxyl or sulfhydryl groups such as amino acids serine, cysteine, threonine and tyrosine. This figure shows the kinetic of irreversible inhibitor. First, irreversible inhibitor form a reversible non-covalent complex with the enzyme. EI means enzyme inhibitor complex. Then this complex react to produce the covalently modified irreversible complex. The rate of irreversible complex is formed is called the inactivation rate or kinate. Binding of irreversible inhibitor can be prevented by competition with either substrate or a second irreversible inhibitor, since formation of enzyme inhibitor may compete with enzyme substrate. In addition, some reversible inhibitors can form irreversible products by binding so tightly to their target enzyme. These tightly binding inhibitors show kinetics similar to the covalent irreversible inhibitors. This figure shows that these inhibitors rapidly bind to the enzymes in a low affinity enzyme inhibitor complex and then undergo a slower rearrangement to a very tightly bound enzyme inhibitor complex. This kinetic behavior is called slow binding. Slow binding often involves conformational changes as the enzyme calms down around the inhibitor molecule. Some examples of these slow binding inhibitors include important drugs such as allopurinol and methotrexate. Reversible inhibitors. Reversible inhibitors bind non-covalently to enzymes, and many different types of inhibition can occur depending on what the inhibitors bind to. The non-covalent interaction between the inhibitors and enzymes include hydrogen bonds, hydrophobic interaction and ionic bonds. Many of these weak bonds combine to produce strong and specific binding. In contrast to the substrate and irreversible inhibitors, reversible inhibitors generally do not undergo chemical reactions when bound to the enzyme and can be easily removed by dilution or dialysis. There are three kinds of reversible inhibitors: competitive, non-competitive or mixed and uncompetitive inhibitors. First, competitive inhibitors. Competitive inhibitors compete with substrate to bind to the enzyme at the same time. The inhibitor has an affinity for the active site of an enzyme where the substrate also binds to. This type of inhibition can be overcome by increasing the concentration of substrate, outcompeting the inhibitor. Competitive inhibitors are often similar in structure to the real substrate. Uncompetitive inhibitors bind to the enzyme at the same time as the enzyme substrate. However, the binding of the inhibitor affects the binding of the substrate and vice versa. This type of inhibition cannot be overcome but can be reduced by increasing the concentration of substrate. The inhibitor usually follows an allosteric effect where it binds to a different sites on the enzyme than the substrate. This binding to an allosteric site changes the conformation of the enzyme so that the affinity of the substrate for the active site is reduced. Non-competitive inhibitors bind to the other side, allosteric sites, not the active sites, and stop the enzyme activity by changing the shape of the active site, caused by disruption to the normal arrangement of hydrogen bonds and weak hydrophobic interaction holding the enzyme molecule together in its 3D shape. This distortion ripples to the active site, making it unsuitable. Therefore, concentration of the substrate is meaningless, unlike in a competitive inhibitor. Few examples of reversible inhibitors. First is acetylcholine esterase inhibitor. It is a chemical that inhibits the enzyme acetylcholine esterase from breaking down acetylcholine. This ultimately leads to increase in both the level and longevity of action of the neurotransmitter acetylcholine. Second, irreversible inhibitor of monoamine oxidase. Monoamine oxidase compromise of a wide range of natural as well as psychiatric drugs that inhibit the enzyme monoamine oxidase temporarily and reversibly. These inhibitors are most commonly used to fight depression and dysthymia.